going to go over in subsequent slides. So these are the two main features of eukaryotic cells. Exchange of information between the inside and the outside. There's a lot of dynamic activity. It's not just something inert and doesn't really do anything in the cell. There's a lot of exchange between the inside and the outside. There is subcellular compartmentalization. And the compartments that you will see will, or the structure that you will see will allow us to infer about the function of the cell. So these are the main two features of an eukaryotic cell. So how does it work, communication between the inside and the outside? There are two ways. One is from outside to inside. This is import. There's another way which is export, from inside to outside. First one is called endocytosis, and it's, for the most part, degradative. It degrades like complex molecules, proteins, etc., into the precursors or the main components or smaller components, and they're used for different activities. The other type is export, and it's being controlled for the most part by the Golgi apparatus that is responsible for sorting components or vesicles out. And that's the secretory path. There is a theory, an evolutionary theory, that has not been demonstrated yet, that is suggesting that an eukaryotic cell has, or the eukaryotic precursor derives from a bacterium, which is smaller. It has DNA without nucleus, without envelope, with ribosomes attached to the membrane. Thousands or millions of years ago, the cell grew in volume that allowed for invaginations here, where the ribosomes remain at the outside of the invagination, that the DNA was protected by this membrane. Little by little, that membrane started surrounding the DNA, the DNA information, and little by little, that became an eukaryotic cell with the nucleus, with the ribosomes outside of the nucleus, with the DNA inside of the nucleus. Then there were other bacteria that were engulfed by this precursor of the eukaryotic cell, and this bacterium that was engulfed was actually of benefit to the cell. So the cell, instead of getting rid of it, decided to incorporate it into new activities or functions. For example, now the bacteria is is well known to be the precursor of the mitochondria, which is the engine of the cell, producing ATP and energy. Okay? So this is how the current eukaryotic cell have evolved from a bacteria. A good reason for a compartment is that a eukaryotic cell is much bigger than a bacterium, so it needs some kind of complexity, structure within the cell. So some reasons for compartmentalization is that there's more membrane, excuse me, more membrane surface to allow more critical membrane activities functions. So there's more invaginations, more membranes that are within the cell, forming, for example, the endoplasmic reticulum, forming the nuclear envelope, etc. The ratio membrane over volume is maintained if you see more invaginations. If you think about it, when the cell grows in volume, if there is no invaginations, no more membrane, it will grow in volume but not in membrane. So that's why the cell needs more membrane inside to sort of catch up with the increase in volume. By having compartments, everything that is produced in the nucleus has a very easy access to the exterior through the endoplasmic reticulum, which we will call ER at this time, Golgi apparatus, and the vesicles that are being sorted to the external surface, or to the plasma membrane and to the exterior of a cell. And as I mentioned before, the evolutionary advantage of the incorporation of the bacterium into the eukaryotic precursor through symbiosis with aerobic or photosynthetic prokaryotes. The existence of compartments means also that, as mentioned before, that there is easy transport from the nucleus to the exterior of the cell. But some organelles, like the mitochondrium, remain isolated from the main traffic. So as you can see here in this electron micrograph, this is now very simple, and the organelles are very highly active, highly condensed here. There's a lot of density of organelles, vesicles of different types, ribosomes, etc. The nucleus, it's highly compact. And there is a direction, which is where we have the information, the DNA in the nucleus. This information is sort of translated somehow, and then there is the production, proteins for the most part, that will execute all the information that is contained in the nucleus. This is just to show you that there's no rule about membrane surface and volume of each particular organelle. For example, some of the organelles, like the ER, has much more surface ratio than the volume. It's highly packed. It's almost all membrane. For example, 60% of membrane here, while it has only 20% of volume. So it has a ratio of 3 to 1 here. It has much more membrane than the volume that it occupies. Yeah.
So in your previous slide, are you saying that information, execution, and production is execution synonymous with translation?
It's right. It's transduction, translation, and many processes. Translation is one of the examples. So you have the DNA that leads to the RNA, that, and then finally to the protein. Translation to the protein. Here we have vesicles, 2%, for example, of surface, 6% of volume. So the ratio, instead of being 3, is 1/3. Some of the organelles here have much more volume than surface. Okay? And it varies. All right, we'll go straight into the nucleus. So these are the main components of the nucleus: the chromatin, the nucleolus, the nucleoplasm, the nuclear envelope, and the nuclear pores. So first of all, the nuclear envelope, as you can see, there is two components: the outer envelope and the inner envelope, and they're not identical. The outer envelope has ribosomes, while the inner envelope doesn't. And that sort of brings back to the origin of the eukaryotic cell, of the nuclear envelope that was formed by invagination of the plasma membrane. The ribosomes have been left at the outside of that invagination, and proof of that is that we have now the ribosomes at the outside. This is connected to the endoplasmic reticulum that has ribosomes. All these dots are ribosomes that are connected, on both sides of the reticulum. But at the nuclear envelope, ribosomes are only existent at the outside of the envelope. Then we have the chromatin here in two main forms. One is densely packed here, which is called the heterochromatin, and it's associated with the inner portion of the nuclear envelope here. And the rest of it, which is the euchromatin, that is a little more relaxed than the heterochromatin, and is the active chromatin. While the other one, the heterochromatin, is packed. It has a lot of histones that make the chromatin very, very packed. Then within the nuclear envelope, we have these pores, which are very important, and they are the nuclear pores. And we'll go into more detail about the nuclear pores. Then we have the nucleolus, responsible for the RNA synthesis, for splicing, and we see some of the functions. The nuclear pores are geometrically distributed throughout the nuclear envelope. They look like they are symmetrical, but in fact they're not. And this is an electron micrograph of the nucleus, showing the nucleolus, the heterochromatin, the euchromatin, and the nuclear pores here with this arrow. So this is the structure of the nuclear pore. It's not symmetrical. It's different from the outside and the inside. It has ring subunits here, but importantly, it contains protein or subunits that are responsible for attaching the nuclear pore to the nuclear envelope. So they anchor all this complex of what is made out of 50 nucleoporins. All this is attached by one subunit. There is a nucleoplasmic ring subunit here. There is a basket at the internal face of the nuclear pore. And the nuclear pore has the capacity to let some substances, some proteins, in or out, depending on how open it is at that time. It has a diaphragm that, or what is called the annulus, that will open or shut this pore, or this channel here, this pore. Some of the proteins that enter the nucleus by free diffusion, the smaller ones, but some bigger ones require active transport. It requires being seen by different set of proteins termed chaperones, etc. And of course, energy, degradation of ATP. So we have an electron micrograph of the nucleus here with the nuclear pore, with the annulus, and with the density packed chromatin or heterochromatin. So this is the structure of the nucleus, of the nuclear pore. This is how it works when there is a large protein that needs to be imported into the nucleus. It's a protein that has an NLS, which is a nuclear recognition signal. I don't quite remember what the other was for. But it's a nuclear signal that is recognized by a set of chaperones called importin, alpha and beta, nuclear localization signal, recognized by alpha and beta importin, that chaperones this protein towards the pore. Then the alpha beta separates, beta stays, and the alpha goes into the nucleus with the rest of the protein because it is attached to the NLS. This process, this translocation process, requires the hydrolysis of ATP and GTP. And when whenever ATP is used, it means that there is a requirement of energy. It's an active process. So a complex protein needs energy to be translocated into the nucleus. And this process is termed gated process through the nuclear pore. It's a very unique way of transportation or translocation of the protein. And once we have the protein inside, the alpha in protein is being released and it's being recycled back outside so that the alpha and beta complex can recognize another protein with NLS. So this is a typical case of gated transport, and it's been involved between the cytosol and the nucleus. It requires chaperone. There are different kinds of protein imports. This is the only case of gated transport into or outside the nucleus. We have co-translational import, which means that at the same time that the protein is being made, it is translocated. For example, at the ER, when the protein is being made at the ribosome. Okay? This process doesn't require chaperones. Finally, there is another case of protein import, which is called post-translational import. And occurs, for example, when the protein comes from the cytosol and gets inside the mitochondria or the peroxisome or other organelle.